Good afternoon, ma'am. Good afternoon, classmates. I am the first reporter, Sherilyn Q. Pelayo, and my topic is all about oxidative pathway, Hexose monophosphate shunt. First, I will introduce my co-members, Danica Bonane, Jonaya Bornalo, Francel Del Socorro, me, Sherilyn Pelayo, and Princess Sanahon. What is hexose monophosphate shunt? The hexose monophosphate shunt, also known as the pentose phosphate pathway, is an alternative pathway of glucose oxidation for the production of NADPH and pentose sugars. So, this hexose monophosphate shunt is a metabolic pathway parallel to glycolysis, and in the TCA cycle, or also known as the Krebs cycle, in the oxidation of glucose. It generates NADPH and pentoses as well as ribose 5-phosphate, a precursor for the synthesis of nucleotides. It has also many other names, like the pentose phosphate pathway, Dickens-Horiker pathway, phosphogluconate pathway, or simply the shunt pathway. It is more anabolic in nature. Anabolic is a metabolic process in which it transforms simple substances into complex molecules, meaning it requires energy to grow and build. And it is not involved in the generation of energy because in this pathway, no ATP is consumed or produced directly, unlike the glycolysis that produces energy in the form of ATP. And it is about 10% of glucose per day is entered in this pathway, while in the liver and RBCs metabolize about 30% of glucose by this pathway. The purpose of this pathway is to reduce NADPH, the production of ribose 5-phosphate or the RP5, and the production of erythrose 4-phosphate or the E4P. So first is to reduce NADPH. The generation of reducing equivalents in the form of NADPH is used in the reductive biosynthesis reactions within cells. So example of this is the fatty acid synthesis. Second is the production of ribose 5-phosphate. This is used in the synthesis of nucleotides and nucleic acids. And the third is the production of erythrose 4-phosphate. This is used in the synthesis of aromatic amino acids. Where is this hexose monophosphate shunt located? The enzymes of HMP shunt are located in the cytosol. It takes place in the cytosol of the cell, the same location as glycolysis. Cytosol is the fluid portion of the cytoplasm. It is the site of intermediary metabolism, the sequence of chemical reactions by which the cell generates and forms the precursors necessary for biosynthesis of the larger molecules used as the building blocks for cell growth and function. The following tissues such as liver, adipose tissue, adrenal gland, RBC, testes, and lactating mammary gland are highly active in HMP pathway. Liver filters all of the blood in the body and breaks down poisonous substances. Adipose tissue is the energy storage. It also provides insulation and protection for organs, and it is primarily made of adipocytes. And the adrenal gland produces hormones that help regulate your metabolism, immune system, blood pressure, response to stress, and other essential functions. While the RBC carries oxygen from our lungs to the rest of our bodies. And the testes are responsible for making testosterone, the primary male sex hormone, and for producing sperm. While the lactating mammary gland, from the word lactating, it produces and secretes milk. So most of these tissues are involved in biosynthesis of fatty acids and steroids, which are dependent on the supply of NADPH. The hexose monophosphate shunt has two phases, oxidative and non-oxidative phases. So in this oxidative phase, it is reversible. The oxidative word of this phase comes from the process of oxidation. Oxidation means the breakdown of a molecule as it loses at least one of its electrons. So during this phase, glucose 6-phosphate is oxidized with generation of 2 moles of NADPH and 1 mole of pentose phosphate with liberation of 1 mole of carbon dioxide. In this phase, two molecules of NADP plus are reduced to NADPH, utilizing the energy from the conversion of glucose 6-phosphate into ribulose 5-phosphate. So in short, it generates NADPH. While in the non-oxidative phase, pentose phosphate is converted to immediates of glycolysis. So, non-oxidative phase is irreversible. It generates precursors for biosynthesis. This non-oxidative phase will be explained further by the next reporter. Step 1. Glucose 6-phosphate dehydrogenase or G6PD is an NADP-dependent enzyme that converts glucose 6-phosphate to 6-phosphogluconolactone. The initial metabolite of the pentose phosphate pathway is glucose 6-phosphate, 2-NADP plus, and H2O. The oxidative phase starts with dehydrogenation at the C1 atom of glucose 6-phosphate, a reaction catalyzed by glucose 6-phosphate dehydrogenase or G6PD. The reaction product is 6-phosphogluconolactone. Conversely, NADP plus is reduced to NADPH during this process. So in this process, NADPH is formed in this reaction and it is a rate-limiting step. Step 2. 6-phosphogluconolactone is hydrolyzed to 6-phosphogluconate by the enzyme gluconolactone hydrolase. NADPH is similar in structure and function as the high-energy electron shuttle NADH mentioned in the cellular respiration. NADPH has an added phosphate group and is used in the cell to donate its electrons just like NADH. Once NADPH has donated its electrons, it is said to be oxidized and is now symbolized as NADP+. NADPH is often used in reactions that build molecules and occurs in a high concentration in the cell so that it is readily available for these types of reactions. Step 3. Dehydrogenate 6-phosphogluconate to 3-keto-6-phosphogluconate, which is spontaneously undergoes decarboxylation to give ribulose 5-phosphate. The oxidative decarboxylation of 6-phosphogluconate by gluconate 6-phosphate dehydrogenase yields 3-keto-6-phosphogluconate, which is converted to ribulose 5-phosphate, a substrate for non-oxidative reactions and NADPH. So the two most important products from this process are the ribose 5-phosphate sugar because this is used to make DNA and RNA and the NADPH molecules, which help with building other molecules. So, to sum it up, oxidative phase starts with glucose 6-phosphate and ends in ribulose 5-phosphate. First is by dehydrogenation. the hydroxyl ion carbon 1 of glucose 6-phosphate turns into a carbonyl, generating a lactone, and in the process, NADPH is generated. Second is by hydrolysis of 6-phosphogluconolactone by special enzyme called lactone. And the third is by oxidative decarboxylation. NADP plus is the electron acceptor, generating another molecule of NADPH, a carbon dioxide, and ribulose 5-phosphate. To explain the non-oxidative phase, here is Jonaya Bornalo.